Welcome to the community webinar for the Gabriella Miller Kids First Pediatric Research Program. I will be your host today. I'm Marcia Fournier. I'm Kids First Working Group Co-Coordinator and Program Director. Today we will have a discussion with guest speakers, and to start and kick off the webinar, I will start with an introduction about the program. The picture you're seeing here is Gabriella Miller. Gabriella was 10 years old when she died from an aggressive type of brain cancer. But before that, she was a fearless advocate. And this program is part of her legacy. In 2014, Congress approved it with a bipartisan bill authorization of Kids First program. And the program was renewed most recently in 2025. The program invests every year $12.6 million to develop a data resource to accelerate pediatric research. The vision of the program is accelerate pediatric research through collaborative research and also supporting data sharing. And we will talk a lot about data sharing here today. I will start, as I said, with an introduction about the program, and then, but the goal here today is to have a conversation involving the NIH, researchers and clinicians about data-driven discoveries for pediatric research. And how it works. When I go to conferences, a lot of patients ask me how patients are connected with the Kids First program. And so I want to show you that all starts with patients and families volunteering for studies with informed consent, participating in studies that are after recruitment by researchers. So researchers recruit patients that consent the use of their samples for genomic sequencing and also allow broad data sharing. And then researchers then bring the proposals to NIH. NIH selected programs prioritizing cohorts that allow that data sharing and also cohorts that bring rich information about phenotypes and clinical information. Once the clinical information, phenotypes, and samples are in, genomic centers that are supported by the Kids First program will generate the data. And then once the data is ready, Kids First also support a data resource center. And the data resource center will compile these genomes, the phenotypes, and the clinical information, and deliver that back to researchers. And now researchers can use that data for accelerating the discoveries. But then after 6 months embargo of this data, the data is also publicly available through the Kids First portal and the dbGaP. So this way now we are sharing this data not only with the original research group, but we are sharing this data to a broader research community, but also to a broader community because this data is available also to patients, foundations, advocacy groups, students, and so in this way is the vision of the program to accelerate discoveries. So to date, we supported 87 cohorts and 37 studies are already made publicly. So not only Kids First helps to generate the data, but also share the data. And today, we are already sharing over 30,000 genomes of affected children and their families. So a lot of our studies have trio or dual design. So not only we have the affected children genomes, but also the families and link it to phenotypic and clinical information, which make this rich data for data analysis. And again, fostering that data-driven discoveries that will help patients affected by these conditions. So in the graph here, I'm showing examples of different conditions that the studies are already publicly released, including pediatric cardiac disease, neuroblastoma, cleft birth defects, Edwin sarcoma, among others. And the bars, what the bars are showing with the different colors is that this data is being assessed over and over again since 2018, showing the relevance and demand for this type of data. I'd like to invite everybody to visit All Studies page, and you can explore. I'm hoping after today you will be curious to explore the data that we're sharing. So please take a look at the study page. Another question I get is why to study congenital anomalies and childhood cancer together? So both birth defects and cancers are leading causes of childhood mortality. In fact, multiple studies suggest that children with birth defects have an increased risk of developing cancer through their lifetime. So Kids First not only generate data across different pediatric conditions, but also different types of data. And what this allows is this cross-pollination. The knowledge can be cross-pollinated and accelerating discoveries. And an example is multiple mutations and drug targets across pediatric conditions that are being reported, that these pediatric conditions have common pathways. So this is important, again, for that collaborative research. And then you may be asking, okay, you generate data, you share data, but so what? How that helps patients? It helps patients through the impact that this data can do. And examples, again, I will invite you to a publication page showing how this data is being used to discoveries in new prognostic markers, treatments, biomarkers, risk assessment, subtyping diseases, which will also have an impact on patient treatment, and condition etiology by having insights on mechanistics, insights on the condition, and knowing more about that condition, again, helping their understanding and also help new discoveries that will help patients. And that's a little bit about the program. And my last thing I'd like to address today is how to assess Kids First data. You can just visit our website, and the website is link will be in the chat box. You can access and explore this data, depending if you're an expert or not. You may be a student, you may be an advocate, you may be a patient. And there is a quick way you can check the data by signing up with your email. We just request that you have a valid email, and in 15 minutes, you can get access to this summary level data, where you can look your gene of interest. If you have a mutation or variant of interest, you can inquire about it and explore the data. If you are more of an expert user, you can request controlled access, and that goes through a process. But because Kids First counts with a dedicated data access committee, this may take few days, not months. It will be less than a week. That is being our experience. Experts now can get controlled data access of individual level genomic data for data analysis. And also our Kids First Data Resource Center, you can leverage a cloud platform, Cavatica. So I will stop here, and this was just a teaser. Now I'm going to invite our guest speaker to join me and continue this conversation. But if you have any questions, please feel free to send us an email. But today here, you can use the Q&A box and start asking your questions. We're going to be answering questions in the end of this. Dr. Sharon Plon, our first speaker. Hi, Sharon. Thank you for being here today.
Happy to be here.
And I would like to introduce Dr. Sharon. She's a full professor at Baylor College of Medicine. She's also a director of the Cancer Genetic Clinic at Texas Children. She's an expert in cancer genetics. And I will start asking Sharon for you just to share with us a little bit more about yourself, your work, how you work with Kids First.
Great. Well, happy to be here. As you mentioned, I'm based in Houston at Baylor College of Medicine and Texas Children's Hospital at the Texas Children's Cancer Center. I'm a medical geneticist. My clinical work is with children and families who have an increased predisposition to childhood cancer. I also run a research lab, and I actually worked with Dr. Lupo on one of those papers that you showed, which was the relationship between the risk of cancer in children with congenital anomalies or birth defects. I actually was in one of the very first batches of the Kids First sequencing projects. We had a clinical trial here funded by NIH, which had children have a clinical exome. So an exome is a simpler test than a genome. And then those parents had consented to research sequencing. And so we actually had the parents, the child and both parents, when available, have genome sequencing through Kids First as a research resource. I now work with Dr. Lupo on one of the Kids First projects related to children with congenital anomalies, and also another Kids First project that we're already analyzing the data related to a specific type of childhood cancer, which is rhabdomyosarcoma.
As you mentioned, you have been involved in studies with both childhood cancer and congenital anomalies. Can you tell a little bit more how Kids First is supporting that type of research, and why is that important?
Yeah, it's interesting because many medical geneticists work with children with congenital anomalies, and a separate group of geneticists tend to work with cancer families or families that have an increased risk of cancer. And what Dr. Lupo first did was combine data. It turns out states keep very careful records of which children have birth defects or congenital anomalies. And they also separately keep very good record of which children are diagnosed with cancer. And so he did a study originally across 7 states. I think we're now across 10 states, where they linked the data from the birth defect registries and the cancer registries. And that was the paper you flashed up. We were able to show that children with congenital anomalies have an increased risk of cancer, particularly children who have more than one. Because I know we have some parents on, I do still want to say the absolute risk of cancer is still very low. It's 1 in 100 or less, but it's more than we see in the general population. So Kids First has played a role because that was all an epidemiology project. We just linked the data, but we only had sequencing on a very small number of patients. It turns out the Children's Oncology Group has a similar survey, and those we have data and we have consent for research samples. And so we now have a Kids First project that's underway where we are sequencing the genomes of children, and if their parents are available, of kids with congenital anomalies and cancer to try to learn what might be causing that. So that's been very exciting to be able to have that data. We had some early data from our own sequencing lab that was able to discover a new condition of girls with multiple congenital anomalies and leukemia. And that's just an example of the type of work you can do if you have whole genomes.
Oh, that is amazing. And so continue on that line of whole genomes. The Kids First data is generating and supporting generation of genomic data, whole genomes. But again, when I go to conference, a lot of patients and families come to me with the question that they go through that process of diagnosis, panel testing, so other types of genetic testing. Can you tell me a little bit about your experience, and also putting Kids First in context of the clinical genomic testing and whole genome testing, etc.?
So I will say, as a busy clinician, I still order lots of panels. Okay, so we know many of the cancer genes. And when a child's first diagnosed with cancer, we're trying to pretty rapidly get a question answered. Is there something in the tumor that might be targetable for treatment? And is there something in the blood or the normal sample that suggests the child was predisposed to cancer? And that can be done quite rapidly and cost effectively by a panel. So I don't think parents should feel like they're being cheated by having a panel. Clinical genomes are now available. They've just become available really in the last couple of years. Ironically, they're used much more for children with congenital anomalies. If we look at the number of cancer genes, cancer predisposition genes, people argue whether it's 300 or 400. If you look at the genes that can cause birth defects or congenital anomalies, it can be 1,000 different genes. So as the number goes up, doing a whole genome becomes more realistic. And so I think we're really in a transition phase. 3 or 4 years from now, will everything be genomes? Maybe. We're getting better at it. We're faster at it. The labs are able to do it more cheaply. But from a purely clinical, a report that goes back to your doctor, there's a great use for panels. The other thing that's probably a little more subtle for parents to understand is when we test the tumor, we're not just looking at DNA, we're actually also looking at RNA. And that really helps with finding things we can target with medications. And so you can do that with genomes, but it takes an additional level of analysis. That's the other reason the parents may be hearing about having panels done, because it's very common for the tumor to have an RNA DNA combined panel. Kids First is a research resource. It is doing genomes for researchers. As you heard, there's an embargo period. It takes a certain amount of time to get the data. It is not a clinical test that's coming out one by one. We send Kids First hundreds of samples at a time. So these are samples we may have collected over the last 10 years. As Dr. Rabin will talk about with Down syndrome ALL, right? So it's being really done for research. It's a very different goal than I've got a new patient in front of me and I need a result in the next week or two.
Thank you. This was really informative, and also the difference between panels, if I understand, if you have a set of target genes is the way to go. But like for congenital anomalies, as you may not have that set of genes already known, the whole genome sequence make a lot of sense. So thank you so much. You will come back,
Okay.
but I want to talk a little bit now with Dr. Karen Braben. How are you today? Thank you so much for joining. And I will start with the same question I asked Dr. Plon. Tell me a little bit about you, your work, and how it connects with Kids First.
Sure. Thank you so much for having me. So I'm a pediatric oncologist, and my main area of interest is leukemia, specifically acute lymphoblastic leukemia, or ALL, which is the most common childhood cancer. And I've had a long time special interest in children with Down syndrome who develop ALL. They have about a 20-fold higher risk of developing ALL. And so understanding why they develop ALL and also how to develop better treatments for them, because they have more complications from their leukemia treatment. I'm at the University of California, San Francisco. I'm the director of the leukemia program there. And I actually was at Texas Children's Hospital for many years until recently, and worked closely with Dr. Lupo and Dr. Plon when they were there. Dr. Plon was my first research mentor, and I'm very grateful for everything I've learned from her. In terms of Kids First, it's been just an amazing resource and opportunity for furthering my research because of my interests just line up so well with the overlap of congenital disorder and a chromosomal disorder and childhood cancer. And I think there's things that we can learn from having a focus on that overlap that there just haven't been opportunities to study in the past. A lot of the genetic sequencing that has been done on a large scale outside of Gabriella Kids First has kind of seen kids with Down syndrome as like a small afterthought, or 2 or 3% of the patients in a study, so they're not really focused on in depth. And we've been able to do sequencing of 200 or 300 samples at a time that are all from children with Down syndrome and ALL to really look in depth at questions relating to that specific population.
Well, that is amazing. And also it's amazing that we have here today, both you and Dr. Plum, which is like the living testimony of the collaborative research that happens at Kids First. You at University of California, San Francisco Children's, collaborating with Texas Children's, and like vice versa. So that is really great to have you here. So I will have a few other questions for you. I think as you're talking about Down syndrome and also understanding the connection between Down syndrome and ALL, tell me a little bit more about that, how Kids First helped you to connect the dots for that. And
Yeah, so we've had a real opportunity with the mission of Kids First to do genetic sequencing. Like Dr. Plon mentioned, it's really powerful to look at the sequencing of both DNA and RNA, to look at both mutations and the expression of genes. And from both the normal cells in kids' bodies and the leukemia cells. And the questions that we have been interested in are partly a number of different things. So one thing is, why do certain kids with Down syndrome develop leukemia? And are there variations in the normal cells in their body that put certain kids at a higher risk? And then also, what is the association, if there is one, with other congenital disorders, and are there certain gene variants that are associated with a risk of leukemia and with a risk of other congenital disorders, and the same kind of pathways are involved in certain structural birth defects and with the leukemia, the changes that lead to leukemia. And then we've also really looked in depth at the genetic changes that occur only in the leukemia cell that drive the cancer, and being able to identify, are there certain changes that we know could be targeted by a certain treatment and could attack only the cells with that genetic mutation and not cause side effects to other cells in the body because we're targeting the treatment to a change in the cancer cell. And also just identifying mutations in a cancer that help us decide how easily it'll respond to treatment, and does it need really high intensity treatment because it's a more resistant type of cancer, or can we do lower intensity chemotherapy that won't cause as many complications because we know it will respond really favorably to the treatment. And then the last piece is we also have some parental samples, and so we can look at we can help figure out like which are inherited changes that are associated with risk of cancer and which are just occurring in the child. And one other thing is the variants in the normal cells in the body affect the way the body metabolizes drugs, and that can give us information about what kind of side effects different patients have, and trying to develop treatments that are adjusted to a patient's risk of side effects and won't cause more serious side effects in the patients who are more sensitive to those drugs.
well, that's amazing. So data can then have an impact on like understanding a patient predisposition to cancer. It can be used to optimize target treatments, assess risk of cancer, for Down syndrome patients, and also assess potential side effects depending on genetic variants. That is amazing. Thank you so much for this enlightening discussions about how data-driven discovery can have impact in so many things that are relevant for patients. So I will now call our third speaker, and then I will bring you back, Karen. And but I would like to introduce Amanda Haddock. She is a co-founder of Dragon Master Initiative and a patient advocacy and mother. Amanda, I will let you introduce yourself, and also how did you become such a data expert?
Sure. Thank you for the opportunity. Well, I got started on this path because my 16-year-old son, David, was diagnosed with an aggressive brain cancer. He was the first person in my immediate family who was diagnosed with cancer, and it really blindsided us. And he was old enough at 16 to ask a lot of questions, which is what we, as his parents, were also doing. And in the process of asking those questions and trying to find the best treatment for him, we realized that they really did not have a lot of great information for us. The questions that we asked were not met with great answers. And we ended up on a path with a clinical trial for him, but we had to choose that clinical trial based on side effects instead of based on what might actually have the best hope of curing him. And ultimately, there was not a cure for David. He passed away at 18, about 20 months after his diagnosis, but we got 20 months with him with a high-grade glioma, and the average back then was somewhere between 9 and 12 months. It's a little longer now. But we never got the answers that we were really seeking for David. And when it became apparent that he was probably not going to make it, he told us that he just wanted us to keep helping other kids. And if you knew David, you would know that other kids meant really all other kids. He would not be satisfied with us just helping kids that, had the same type of tumor that he had. And that was tough for me in the beginning because he had, at the time, they were calling it glioblastoma. And so that was really my very narrow focus. And we set out on a path to try to cure glioblastoma. But almost immediately, it became apparent that we knew so little about that disease that we really needed to look at other types of disease to try to find some additional clues. And that's kind of what the researchers were telling us as well. We just kept hearing that they needed more data, they needed to understand, they needed to be able to look at more patients. And very quickly, we were starting to hear that they needed not just cancer patients, right? That we were starting to see these little signs of, children with these congenital anomalies were getting cancer at a higher rate, and no one really understood why. And so combining the data and being able to look at that together really made a lot of sense to us. So we got involved with data collection. There wasn't like back then data sharing needed data collected first. And so we were involved in data collection first in the genomic space and helped develop a tool that Kids First uses called Cavatica. And then from there, we really started with the data sharing part. Now that there was some data collected, how do we get it into as many hands as possible? And that was not a very popular thought. We started this nonprofit back in 2013, and people told us we were crazy and that was never going to happen. And they were not, researchers were not going to share that data. And fast forward to now that it is not a super common practice, but definitely something, thanks to efforts like the Kids First Data Resource Center, there's now places to put the data. So there's a good reason for people to share it. Before there's no place for the data to go. And so now there is a place for the data to go, and there are people looking to use that data. And so hopefully going to be commonplace very soon.
And I have a few other questions here for you. So I will go again and touch on the cross-disease approach that Kids First has taken. Why is it important to have data from different conditions in one place and being able to work together? So can you tell a little bit like why we should care about that type of approach?
Yeah, I feel very passionately about that type of approach, so I'm happy to discuss that. So I think the more we learn, right, about cancer and the mutations that are driving its growth, we see that those mutations happen in a lot of different types of cancer, and they're happening in other types of disease as well. And so if we're only looking at cancer based on the body part that it occurs in, then we're missing the chance to utilize information that may have been discovered in another body part, right? And so if we're looking at, BRAF mutations, very common, people hear about that a lot, but they may have made a lot of great discoveries in the breast cancer world. And if we're not looking outside just brain cancer, then we'll never see those other discoveries. And the thing that I like about Kids First in particular is, there is that 6 months that we have to wait until the data is open, but that's better than the years and years that we're having to wait out in the real world, right? Most of the time, data does not get published until after a paper is published, and that process can take easily 10 years. And our kids just don't have that kind of time to wait. We really have to get this data out more in a more expedient fashion so that we can make discoveries more quickly.
Thank you. And I think I'm gonna now, I would like to invite all our speakers to join as a panel. And I'll have a couple of questions for you all, but I see a lot of questions already coming through the chat and Q&A. So we will open soon. So if you have questions, please continue adding your questions to the Q&A. I'm gonna look at that very quickly. I think one thing that was all three of you touched is on this, the Kids First is helping with the research, whole genome sequencing and data sharing. But there is also patients are interested in learning more about genetic testing in a clinical setting. So I'd like maybe all of you commenting a little bit. Oh, you may say, okay, so it's because this research doesn't have value. So how this research data is bringing value for patients that are like now trying to going through the process, going through the treatments and diagnosis process of their own journeys.
Well, I can start. I think it's important to realize how rapidly genetic testing labs can add genes to their panels. So for example, as I mentioned, we use genome data to identify a new leukemia susceptibility gene, which we published. And actually we had parents reach out to us even just from the publication. And Amanda, I did just want to say I'm very sorry for the loss of your son. And thank you for sharing that. But labs now have already added that gene to their leukemia panels. So, research gets published, clinical labs pick it up actually very rapidly. And so I don't think parents should feel like, oh, it's only research. These two worlds are in sort of constant communication. I'm sure Karen can give many examples in leukemia, how now every patient with leukemia gets certain things looked at that were a research study a few years ago. So, the research data is being published so that it can be taken up by clinicians and clinical labs.
Yeah, I'd be happy to chime in. As Sharon said, I think oncology is a field where the research makes its way very quickly into clinical use, and the use of sequencing data has become really standard in a way that wasn't true just a few years ago. I'm a part of the leadership of the ALL trials by the Children's Oncology Group, which runs clinical trials for treatment of childhood cancers in over 200 sites and most of the cancer centers in the United States. And the next trial for standard newly diagnosed ALL will be using both DNA and RNA sequencing data to determine the risk group for patients, because there are certain risk groups that we've identified on a research basis in the past 5 years that we know are either better risk or higher risk and need to have their treatment adjusted for that reason. And it's already being implemented in the next year or so in the next clinical trial. And then there's also some really nice work at St. Jude that has identified a new gene that's involved in the metabolism of one of our common oral chemotherapy drugs called mercaptopurine. And we make adjustments. We now test clinically for how patients for that gene, NED-T15, if that enzyme works not as well and leads to more side effects from the use of mercaptopurine, and we make adjustments to their dose of that drug based on that knowledge. And that, again, is something we've just figured out in the past few years as another gene that we need to know about to give the right doses of our chemotherapy. So a lot of examples, and there are some specific genetic changes in specifically in kids with Down syndrome that drive their leukemia that we have a much better understanding of now than we used to, and that there are some targeted therapies that are being developed that may help for those cases. So it's really an exciting time to see all the progress that's coming year by year.
No,
And-
yeah, it is amazing to hear from you how faster now it is the process of incorporating this new knowledge and new discoveries you can quickly maybe adapt and incorporate quickly in to benefit patients. So I'm really impressed by hearing how this is now so much faster than it used to be before. Amanda, you want to comment on?
I mean, I guess I would just encourage parents to really consider being involved in research like this, because I think sometimes when you're in the throes of trying to find a cure for your child, this might not seem critically important, but I think both doctors really spoke about the pace of research and how, to me, it just seems like it's exponentially faster, right? In the like 12 or 13 years that we've been doing this now, it's just amazing to me how much more quickly things are moving. And so you might think, oh, I'm just doing that for research and it's never going to help my child. But I think that we're getting very quickly to a point where we will be translating those discoveries even more quickly into care. And so I think it's a little short-sighted to think that it might not help your child. It might not, but it also very well could. And so I think that I think very few people would feel badly about helping research move forward that helped a child that came later, right? That's always something that you will be able to do with research. And so I know for my son, he really felt strongly that that could be part of his legacy, no matter how it ended for him, that he was always going to be pushing research forward, and it was always going to matter for somebody, whether or not that somebody was him.
And I'm really so humbled to be on the panel with you and by the work you're doing, Amanda. And it really is the knowledge of our patients and the desire to help them and to make a difference for them that drives all of this. And it's
Great.
just amazing the questions you ask and the work you're doing to try and move that forward. Thank you.
Thank you.
Yeah, thank you, all you three. And I will, as I promised, I will look at now our audience questions. But before going to the audience questions, I want to have for Amanda, for the parents that are watching right now and maybe overwhelmed with the process that they're going through. What is your advice to them? And then for Dr. Plum and Raven, what is your advice to other researchers and clinicians that alluding to do some work like that with Kids First?
I think I would just really encourage everyone to pay attention to this kind of a project because it can be so overwhelming when your child's diagnosed and very isolating. And taking your child's information from sometimes really an N of one sort of position, right? When we're looking at all the different conditions that are covered in Kids First, maybe you don't have a community around your child's diagnosis. But by putting their data into an environment like this, you now have a community, right? And there is now this whole extra set of researchers who are able to access that data and look for new discoveries around your child's condition. And I think it's such an amazing time for us to be able to do that, right? Like before, you would have just remained in isolation, and now you've got this, unique diagnosis that no one knows what to do with. And by putting that child's data here, almost immediately they start to draw connections between other types of diseases. And that gives me a lot of hope for the future and a lot of hope for these children that we're going to be saving more and more lives.
Thank you. And I think I just want to comment on this sentiment of isolation, also reflecting on rare diseases for researchers also. Sometimes researchers will have a cohort of 10, but now with Kids First, this will be in a context with 30,000 other genomes that it can relate. So that is also that aspect that I want to highlight. So Dr. Plon, do you want to comment on advice for researchers and clinicians that are thinking of pursuing this type of
Sure,
data-driven?
sure. And I can be brief because I see there's several questions in the chat. I do just want to emphasize what Amanda did, which is data sharing. So to be honest, I think the genomicists were out ahead, right? Clinical genetics has had a requirement for data sharing for 15 years already, which NIH has now instituted across all of NIH. And I think that the idea that you should hold on to your data generally just slows you down and slows the community down. So I really strongly recommend data sharing. If we had time, I can give many, many examples where sharing data very early helped my own lab, not just other labs. And so, I think that's critical. I think for clinicians, they are very busy people, but I think there's more and more effort to be able to use data in the medical record as part of research. And so sort of joining in on some of those efforts and thinking about ways you could structure your note to be easier to be used by clinicians and by researchers. And not viewing that as a hindrance, but maybe a way to help your own clinical practice at the same time that you're helping researchers is something I would recommend.
Yeah, just briefly, I think Amanda and Sharon have said a lot on this area, but I think what is really the most powerful is the sharing and collaboration and communication back and forth between clinicians and researchers to enrich the data and learn more from it. And researchers have the genomic tools and new ways of analyzing data and obtaining data. And then the clinicians have, the important key clinical questions to go after. And I think a huge part of the culture in pediatric oncology is enrolling patients on clinical trials. And that's a way that the clinicians generate the data about both collecting samples from those patients and really detailed clinical data of how they're responding to the treatment regimens. So then we can draw correlations between the genetics and the outcomes clinically. And that's the most powerful, or a very powerful form of research that's really a collaboration of the two groups.
Thank you. Thank you, Tri, for being here today. I will now move to the questions from the audience, but just as a recap, we learned today how data research can impact in so many aspects that relevant for patients like disease predisposition, target treatments, risk assessment, assessment of side effects. Also, how research data can be now fast incorporated in the clinical practice in a much more dynamic way that we ever imagined before. And also the data sharing, also participation of patients so central to all of this, and building this community. And I'm really humble that we are supporting this type of projects. Thank you so much for all your work. And I'm looking forward to continue our collaboration. And I'm gonna now start reading here some of the questions. So we have a question from Hiren Kuratia for Dr. Plum. We are currently transitioning from panel to clinical genome. During this process, effective communication between researchers and clinicians is essential to bridge the gap between data-driven discoveries and understanding genotype, phenotypes, causalities. I would appreciate your thoughts on how we can improve collaboration communication to help this transition.
Well, that's a big transition. I think I would first just remind you that it's a clinical test, and your first duty is to provide the clinicians the information they need. And that can be really challenging with genomes, is to figure out the best way to report it so the clinician understands the results and understands which results are the most important ones for the patient. I think then, as a secondary thing, many labs do it different ways. Some labs have, as part of their consent form, the parent agreeing to have the data also used for research. Some have a process by which they reach back out to the clinician and have the clinician talk with the family about using the data for research. And so that, I think, is really done differently by different labs. Often clinical labs also share their data on ClinVar, and that is a database that's used widely by both clinicians and researchers. So I think there are many ways in which you can take that clinical genome and use it as a research tool. But I think your first duty is really to figure out the best way to report the genome finding to the clinician who ordered the test. And then then the research connection is secondary.
We have a question from an anonymous attendee. What techniques are commonly used to identify genomic mutations during clinical genome?
Yeah, and I'll just be brief. There's a very large number of tools, many of them developed originally by the genome centers, that are now available to diagnostic labs. There are actually many commercial pipelines that many of the big sequencing companies provide. And so each clinical lab, and again, these are people who are board certified in clinical testing, will decide which of these pipelines is the best to call mutations that they're going to report to patients.
Before I go to the next question, Karen, do you want to also address a little bit, have any addition to techniques that are commonly used?
No, I think the basic, Dr. Plum has provided the basic response.
Great, thank you. And so now a question for myself. There being any talks in coordination between the NIH All of Us program, which is now enrolling young children. All of Us program is similar to it, collects genomic samples and qualitative data and makes it accessible to researchers, not limited to cancer. Yes, we Kids First and All of Us, these are both programs part of a bigger umbrella of the Common Fund data ecosystem at NIH. So we're very excited now. It's actually all of us, including children, will be recruited. And I think we're looking forward to collaborate with the All of Us program. One key difference that I want to highlight highlight that distinct is all of us program is recruiting all individuals, and Kids First, our data sets are related to, there is some biology behind some condition or diseases, and also as opposed to only having individual data, we also bring family data. So there is some distinctions, but nevertheless, very synergistic, I think, opportunities among the two programs.
I would just
Dr.
add that childhood cancer is very rare. And so even in all of us, right, so we're doing a project right now with like 800 rhabdomyosarcomas in Kids First. And in all of us, there's probably 20 or 30. So I think all of us is a great example of a population data where we have the health record. But Kids First really is designed for cohorts, right? We have to apply as a cohort, like Karen talked about Down syndrome, ALL, right? And so Kids First allows you to really zoom in on one disease, and all of us lets you look in the bigger population of what that gene looks like.
So I think a little bit about like, how is your process to recruit patients to participate in your studies? Can you, just expand a little bit on what is the process of recruiting patients for a cohort, for instance, for Kids First, so people understand?
So Karen, do you want to take that?
Sure. So a lot of our recruitment is for children with cancer is through the Children's Oncology Group, and there's a what's called an umbrella protocol called Project Every Child, which is not related to a specific treatment study, but for collecting clinical data and samples, and a lot of our families participate in that just kind of out of a spirit of altruism, because that isn't necessarily information that definitely will help their child, although as Amanda said, it may, but they want to, even in this time where they're going through something so challenging, they want to be able to have that experience help others, and so that's really an amazing gift that we're grateful for. Then there are also clinical trials for the treatment of cancers, and often those include submitting research data and samples, and so we learn how we're able to study samples that are associated with a specific consistent treatment for all of the participants. So that's very powerful data as well. And then we have some studies that we approach patients in clinics specifically for children with Down syndrome, healthy children who don't have cancer, and that's an important resource, because all of this, we need to know how do things compare between children who do develop cancer and those who don't.
Actually, could I add something there too? Because Dr. Raven said something important. I think we do a really great job of collecting data on kids that are on studies or on a clinical trial. But that's just not always the journey that families choose, right? Especially with incredibly aggressive cancers. And I think there's a real opportunity here for people who are not choosing to go on a clinical trial to still opt in and be part of research, and to have their child's data studied. And the same that you just mentioned, as children with Down syndrome that are otherwise healthy. It's important to have data on these children who aren't going through the clinical trials, who aren't getting those treatments. So if that's the decision that a family makes, I think that they can still be a really important part of research by opting in for a study like this that just tracks that information and lets us see how those children do that don't go on these trials. That can also be really valuable information. And there's not always an easy path to doing that. Kids First gives them a really easy way to connect with their physicians around that.
This is such a good conversation. I could continue on and on, but I would like to finish with a touch on the future. Tell me a little bit what excites you about the future, and what something that gives parents a reason for optimism. I see here on the chat, actually, I will share this question to the whole panel. What is something that gives optimism for parents and your future vision of where we're going with data-driven research.
Can I go first on that?
Yes.
So I think that the data collection that's been happening through Kids First, it was really visionary. And we're all seeing, obviously AI is touching every area of our life, but AI is only as good as the data that you have to drive it, right? And so Kids First has very high quality data that is poised to be able to interact with these AI tools in ways that can really drive research and care forward in ways that I think we're only beginning to explore right now. So I think the future is going to be amazing with how we're using this data for kids and adults.
Yeah, so I would say just briefly, again, so the capacity to collect all different forms of genomic and proteomic and metabolomic, just all of this different type of data and the computer computational resources to integrate it and learn from it is really exciting. And then as an oncologist, I have to say it's just so exciting to live in this era of immunotherapies and being able to harness the body's own immune system to attack the cancer rather than relying on toxic drugs. And there's a lot we can learn in the genetic world to help optimize that therapy, but that's been just a transforming thing in our therapy these days that really it gives a lot of hope and a lot of benefit for curing cancers we didn't used to be able to, or doing better at curing them.
I actually was going to answer in a little different way. If you are a parent of a tumor we're still not doing that well at, I know it can be frustrating to hear these stories. There are a lot of breakthroughs, but we still lose too many children to cancer. And I think the work of research is to try to reduce that number every year. But and participating in research, as we heard so beautifully from Amanda, may not have helped her son, but could help someone in the future. And that can be gratifying to a family that otherwise it doesn't have a great option right now.
Thank you all for being here today. It was really an honor talking to you. For Kids First, I think for me, what excites me in the future is that growing this data resource, parents will be able to be empowered by this information, even to understand better about their conditions, but also to be part of a community, as Amanda said, not feeling lonely, and being part of something. So that I think is excites me about the future of Kids First and working with you all. And thank you for the audience for being here today. And see you next time.